Here we have a starter question based on the light reactions, which was our last PowerPoint. In this question, you are being asked to identify the name of the first stage using the word bank. So for number 1, you're needing to identify the name of the first stage of photosynthesis. In area number 2, you need to identify the 2 products that are used in the second stage. And for number 3, the product that diffuses out of the leaf. You have to use the word bank to support you with your answers. We should have identified that number 1 is the light reactions, that's the name of the first stage. Number 2, ATP and hydrogen are the products that are used in the second stage. And number 3, we should have identified oxygen as being the product that diffuses out of the leaf. In this PowerPoint, we're going to focus on 5 learning intentions from page 14 of your student guide. We're going to be able to, hopefully by the end of this lesson, be able to name the second stage of photosynthesis, identify the raw materials of the second stage of photosynthesis, identify the products, describe the events that occur during the second stage, and finally be able to name the storage and structural carbohydrate produced from glucose. If you click on the image appearing on the screen in front of you, you'll find that this video is hyperlinked to an overview of the whole process of photosynthesis. We did watch it in the last lesson. It might be worthwhile watching it again to recap the light reactions and to give you an introduction into the process of carbon fixation. If you click on the image and watch the video, please remember to come back to the presentation to continue with the next few slides. We've already seen that aspects of photosynthesis link with our prior learning. It's important that you are familiar with leaf structure, that you are important with cell structure to include chloroplasts and chlorophyll, and we will be mentioning the likes of respiration and enzymes in the remainder of this PowerPoint. So if you're unsure of any of these aspects, it would be beneficial to perhaps go back to the appropriate part in a student guide or on Teams and watch the narrated PowerPoints again. In terms of a recap from our previous lesson and a full overview that we've had on photosynthesis so far, we have already discovered that photosynthesis is an enzyme-controlled process by which green plants make their own food in the form of sugar. We have what's called a summary word equation for photosynthesis, where we look at carbon dioxide and water being identified as what we call the raw materials. Light energy that's absorbed by chlorophyll helps to produce sugar and oxygen. In the last PowerPoint and the last set of teaching, we had looked at what happens in the light reactions, which involved the chlorophyll absorbing the light energy, that then being used to split a water molecule, which formed oxygen as a byproduct. We've also looked at the process of photosynthesis being 2 stages. Stage 1, the light reactions we looked at in our previous PowerPoint. And in this PowerPoint, we're going to be looking at the process of carbon fixation. Carbon fixation is highlighted on the screen in front of you, and in class you would receive that as a handout. Carbon fixation is the second stage of photosynthesis. It occurs in the chloroplast of a cell. Carbon fixation consists of several enzyme-controlled reactions. Take your time to make sure that you have noted down this key learning point. The second stage of photosynthesis, being controlled by enzymes, sees the form of a cycle that results in carbon, which comes from the carbon dioxide, becoming fixed into a sugar, the main sugar being glucose. How this fixing or formation of the sugar comes about is because the carbon and the carbon dioxide gets combined with the hydrogen and the ATP from the light-dependent stage. So a series of enzymes are involved in combining the hydrogen and ATP from stage 1 with carbon dioxide to produce sugar. For carbon fixation, it's important that you're able to identify the raw materials, the products, and essential requirements for this stage. The raw material for carbon fixation is carbon dioxide. The product is sugar. And because carbon fixation is controlled by enzymes, an essential requirement for carbon fixation is temperature, because all enzymes operate at an optimum temperature. The diagram on the screen in front of you is showing the effect that temperature has on the rate of photosynthesis. Now, we have identified that carbon fixation is controlled by enzymes. Enzymes, linking our learning back to cell biology, all enzymes are affected by temperature. So therefore, if enzymes control carbon fixation and the rate of photosynthesis will ultimately be affected by temperature then. If we have a look at the graph, the rate of photosynthesis is increasing as the temperature increases. But we can see that the rate of photosynthesis reaches what we call the optimum. That's the best working temperature for the enzymes that control carbon fixation. The rate of photosynthesis will now start to decrease as the temperature increases further above the optimum. We can actually see that the rate of photosynthesis drops to 0 because at that point the enzymes that are controlling carbon fixation have become denatured due to the high temperature. As a plant grows, it continues to make glucose by photosynthesis. Some of this sugar is broken down again by respiration to supply the plant with energy for vital processes such as cell division and reproduction. Some of the remaining glucose molecules become linked into long chains and packed together into starch grains found in the cell's cytoplasm. This plant product is the plant's store of food and can be converted back to sugar when energy is required. Starch is therefore called a storage carbohydrate. Other glucose molecules are built into long chains of cellulose. These are gathered together to form the fibres needed to build cell walls. Cellulose is therefore called a structural carbohydrate. Here we have some assessment standard questions for carbon fixation. There is also a crossover to the light reactions in the first question, and that's a wee bit of a recap from our previous lessons. The first one's about applying your knowledge at a level C. You're being asked here for the 4 statements to identify whether they happen at stage 1 or stage 2. The second question is a level A, so there's a little bit of depth of knowledge required, a little bit more challenging. And you've been asked to explain why high temperatures above 50 degrees would prevent the photosynthesis reactions from taking place. Give you a few moments to have a think about these, and then the answers will be revealed. Water is required for stage 1. Sugar is produced in stage 2. ATP and hydrogen required is for stage 2, and oxygen produced is stage 1. Why temperatures above 50 degrees would prevent photosynthesis reactions from taking place is because photosynthesis is controlled by enzymes, that's for 1 mark, and then these enzymes would be denatured at the high temperatures for your 2nd mark. That's us completed the PowerPoint for carbon fixation. You are asked here to reflect upon your own learning, evaluate your own learning, and if needed, go back to the appropriate slide to revisit the area that is still causing you some concern.